So the first topic were what are mechanistic updates and new approaches. And there have been a large number of in vitro studies and animal studies. And the question is, what non-thermal molecular and cellular changes are reproducible? Well, in terms of the animal studies, the correlation, consistency between the tumors seen from the NTP study of glioma and Schwann cell tumors appear to be consistent with what has been reported for human studies. Ramazzini studies are still awaiting a PWG. Ramazzini, I think Fiorella mentioned, was the group that first identified vinyl chloride as a human carcinogen, or a carcinogen in animals with angiosarcomas of the liver, which...
By number of chemicals, we are second to your lab, to the
laboratory. Right. It's huge.
Chemicals evaluated in the world next to the U.S. government.
Yes.
Right. Okay, but a lot of the important chemicals, trichloroethylene, benzene, NTP, and Ramazzini came out around the same time. In fact, that was kind of strange that there was human evidence, but at that time there wasn't animal evidence, so people were concluding, well, it can't be a human carcinogen because we don't have the animal evidence. But so with respect to findings that are reproducible on the animal studies, we're still awaiting the Ramazzini. But in vitro studies, the strongest consistency appears to be for oxidative stress, cellular stress kinases, some cell cycle change, and altered gene and protein expression. However, there's this issue of inconsistency, and it still exists for all endpoints that have been measured, and this has become then an argument against mechanistic studies. There are so many variables that you can introduce into mechanistic studies that these may account for the inconsistency itself. For example, the different strains of animals, the species, for in vitro studies, if it's tissue or cell type, the sensitivity of the cells to the RF radiation can be different, and therefore there's a need to replicate this under the exact same conditions. Exposure intensity or duration of exposure, these vary from study to study. Some studies will expose for short term on an in vitro, some will expose longer. As I tried to explain on the NTP studies, we thought they were all the previous studies were underexposed. That's why we extended it. So exposure intensity and duration are important. Exposure systems vary for in vitro as well as in vivo. Depends on the analytical methods that are used that may have different sensitivities. Measurement of dosimetry can vary from study to study so that we're not comparing actual dose to dose, or maybe inadequate control of temperature. So what are we going to do about all this? This then becomes some areas that we think would help for further research. We need to resolve inconsistency among the mechanistic studies for the same endpoint, such as oxidative stress, heat shock proteins. How can we do this? Well, one possibility is swap between laboratories that report a positive result with those that report a negative result for the same endpoint. However, under the conditions of findings of oxidative stress, since the IARC 2011, there have been a large number of studies in vivo and in vitro, and they're not all positive, but something like 75% have been shown to be positive. The fact that we now have DNA damage in rat and mouse models, we have a possible mechanism of oxidative stress. One suggestion here is to see if we can evaluate that potential relationship between oxidative stress and DNA damage in rat or mouse models. Because once you start working on a hypothesis of a pathway towards carcinogenesis, this then provides that mechanistic linkage between exposure and the outcome of tumorigenicity. And with that, that then becomes in addition to what you mentioned in terms of evaluating the genomics of the brain cancers, another approach is within the animal models. If you can show that we can explain how tumors might have occurred, that provides then strong mechanistic evidence. With strong mechanistic evidence, sufficient in animals and limited in humans, would still be a known human carcinogen. A suggestion by one of the organizers here was, does magnetite predispose RF exposed children to autism spectrum disorders, an effect in vitro mechanistic studies, and to evaluate the synergistic effects with magnetite from air pollution. So the relationship, can magnetite be a synergistic factor in creating autistic spectrum disorders? And another was to evaluate gene and oncogene tumor suppressor gene mutations in brain tumors that have been induced by RF in rats. And some of the genes to look at were considered isocitrate dehydrogenase and fumarate dehydrogenase. And then some of the more conventional proto-oncogenes or tumor suppressor genes, such as KRAS, P53, P16. We also asked the question, how can mechanistic studies address differences in susceptibility with age and underlying variabilities? The approach that we considered is to compare effects such as the induction of reactive oxygen species or male fertility after exposure at different life stages, such as from in utero exposure, lactational exposure, prepuberty, and adult, to address the issue of other underlying variabilities. There are a variety of knockout mouse models that might be considered. So, for example, the enzyme 8-oxo-G DNA glycosylase. This is a base excision repair enzyme that removes the 8-hydroxydeoxyguanosine addict formed by reactive oxygen species. There are also other means of silencing genes or addition of antioxidants, if we can show antioxidant susceptibility, reducing the oxidative stress. I think that adds stronger evidence for a pathway or mechanism of events. How can we identify and validate potential biomarkers of exposure? The approach that we are recommending are metabolomics. This would be looking for metabolites that are produced in, could be in animal models or looking in humans, of the urine, saliva, exhaled breath from animals at different ages. In animals, there's a lot less variability in terms of dealing with additional exposures that might impact metabolomics. Metabolomics is basically looking at physiological changes in which metabolism is altered as a consequence of perhaps a stress related to RF exposure, and the general metabolites that are produced show some type of alteration. If there's a key altered metabolite, that would help us identify a potential biomarker. Then there's also biomarkers of response. These would include analyses of proteomics, altered gene expression, gene mutation in circulating serum DNA, which came up as a suggestion as well. What are the most important co-exposures for further research? That is, toxicants, beneficial compounds such as nutrients. One that, after we heard a lot about that 5G that's being rolled out in the next couple of years, and the effect of it on the skin, the damage that it might create in combination with UV light might be a synergistic response. The addition of gamma radiation, as has been used in the Ramazzini studies, is obviously another co-exposure. Hazardous air pollutants, a number of these can also be evaluated in conjunction with RF exposure. And in terms of nutrients, dietary antioxidants, how do they influence the outcome with specific endpoints such as oxidative stress? Yeah. Yeah.
What about heavy metals?
That would probably be under the hazardous air pollutants.
The question was asked about
Metals.
Yeah, they were-
Hazard. Heavy metals as well, yeah. Yeah. We're not limiting it to just what we have here, any type of, metals for sure. What is the biological basis for medical uses of radiofrequency radiation? We heard about the use of this for treatment of glioma using, I forgot the frequency.
Tumor treatment field.
Tumor treatment field, okay. And the underlying mechanism there appeared to be inducing apoptosis in dividing tumor cells. Alternative basis for potential use would be antibody targeting with an RF absorbing conjugate, such that with that kind of labeling, then exposing to RF might be able to target specifically where that antibody binds. And there's been a number of studies on altered blood-brain permeability, such that this could be, in some cases, bad, obviously, if you're exposed to a toxic, but in some cases it may be useful also for drug delivery by altering the blood-brain barrier to get chemotherapeutic drugs to brain for brain cancers. What studies exist of newer technologies such as 4G and 5G? Okay. We feel that this is an important issue and that there is an urgent need to evaluate health effects, including the skin physiology of 5G before it is implemented and people are exposed. Here we are going again into a new technology without any knowledge of what this might mean on health effects. What we know so far at this time are that the sweat ducts are very strong absorbers of 5G radiation, but we're not aware of any adequate health effect studies on 4G or 5G. So we need to evaluate if 5G, for example, increases the risk of skin diseases such as melanoma or epithelioma. And we also believe there's a need to examine brains of rodents exposed to RF in utero during lactation for dendrite outgrowth, since this may be linked to the autistic syndrome. If we can create a evaluation of potential relationships between early mechanistic events and precancerous events, we then are on course for a pathway that leads from exposure to tumorigenicity. The precancer events may be genetic or epigenetic alterations. There's been studies, positive and negative, on altered blood-brain barrier. This, I think, is necessary that this be confirmed. There's lack of any experimental data, it seems, on health effects of Wi-Fi, and it's obviously a substantial need. Relationships between reactions to stress and altered cell signaling, and all the other data gaps that we mentioned above, we feel need to be addressed.